Hello, and how are you? Welcome to our session on cell organelle again. This is part 2, and it's a continuation of the previous class that we began on cell organelles. So we checked on other parts of the organelles, and now we'll be doing the second part so that we can complete the remaining bits. If you're new here, my name is Winnie Barawa. Stay with me till the end as we discuss more on the cell. So in the previous session, we did the first 6 organelles. So we checked on their features, we checked on their functions, and we saw also the processes they do in their various locations in the cell. We've also explored on how the cell has been organized, from how systematic the cell is with regards to the different functions that our organelles operate on. So in this class, we're going to handle the organelles from point 7 here all the way to number 14. So we're going to do all these organelles, looking at their features, characteristics, and so continuing with the lysosomes. So by structure, lysosomes are organelles that are known for containing lytic enzymes. They are bound by a membrane, and their main function is to break down food, waste, and damaged cells all within damages all within the cell. So they help us clear any material that is worn out or is not needed anymore in our cell. So how does the lysosome do this? So the lysosome is able to establish this using different methods. For example, the lysosome can digest particles taken in by endocytosis, specifically ingested viruses and also toxins. And the other way of destroying what the lysosome does is it degrades any worn out or non-functional organelles. The lysosome also performs metabolic functions, including glycogen breakdown and release. It has other functions, including breaking down non-useful tissues, such as the webs between the fingers and toes of a developing fetus and the uterine lining during menstruation. And the last bit is breaking down bone to release calcium into our blood. So looking at the metabolic functions of our lysosomes, being the fact that its main focus is breaking down waste materials, toxins, or damaged products, it will do different things based on the location that it is. For example, within the cell, there's endocytosis that happens, and in this, with the endocytosis, we're able to destroy any form of viruses or toxins. While when it comes to non-functioning organelles, just still within the cell, it can operate on that also. And the other bit is on the breakdown of glycogen, breaking down of non-useful tissues in the case of embryology, where we have the spaces in between the fingers or the webs. These are also being destroyed again by the lysosomes. And the functions are many, just to repeat, based on where our lysosomes are located. Next organelle is what we call peroxisome. So the peroxisome, again, it contains enzymes and it is also membrane bound, just like the lysosome. But the main function of peroxisome is to protect the cell from any form of toxins, although the main focus of the toxin that we're talking about here is the hydrogen peroxide. We both know hydrogen peroxide is very dangerous because it can react with the organelles and the fluid in our cell, that is the cytoplasm. And for this purpose, to keep it safe, the peroxisome has that function of protecting us from this hydrogen peroxide. So peroxisomes are membranous sacs that contain several types of enzyme, but most importantly contains what we call oxidases and catalyses. So what the oxidases use, the oxidase uses molecular oxygen to detoxify harmful substances, including the cases of alcohol and formaldehyde. So with this containing parts of oxidases and catalyses as our enzymes, then this peroxisome now gives us the chances that it can oxidize or it can catalyze different substances by the use of oxygen for the purpose of detoxification. So it can literally damage or it can literally detoxify or bring down the severity of the toxins that are within our cells. And remember, as we break down different substances within the cell using the metabolism, we therefore generate also toxins at this particular moment. So for us to keep our cell safe from such toxins, then peroxisome comes in to destroy any forms that are coming in as alcohols, the peroxides, or the formaldehydes. And the other organelle is the vacuole. So the vacuole is also a sac-filled sac. So here it is, the green substance here, the green organelle. And the main function of the vacuole, it helps to store materials for the cell. And to remind us about the Golgi apparatus, remember the Golgi apparatus had a cis phase and it has a trans phase. And in all these phases, we used to have the vacuoles coming in with the materials and transporting them to the other needed areas. So the vacuoles can work together with the Golgi apparatus, but they work closely also with what we call the vesicles to transport materials. The vacuoles are very good at storage, even though they also actively participate in the transportation, but the main agenda is to store materials for the cell, whether needed for different functions or needed for other parts of the cell. Next is our famous mitochondria. So the mitochondria, by shape or by structure, it is bean-shaped. It is double membraned, so it has two membranes, and it has its own DNA and ribosome. So let's check on our structure here. So we're saying the mitochondria is bean-shaped, okay, and it has two membranes. So it has the outer membrane and it has the inner membrane. So the interesting part is that the inner membrane has projections, and this projection is what we call cristae. And this projection is very key because it helps in increasing surface area for the purpose of energy generation. So with the cristae, to increase the surface area, we have the matrix inside the mitochondria, and the matrix is the site for the cellular mechanisms or for the cellular respirations. But the beauty about the mitochondria is that you are told it has its own DNA and it has its own ribosomes. So what are the functions of the mitochondria? The mitochondria works to supply energy to the cell, and it is the site of cellular respiration, or rather it's the powerhouse of the cell, because that is where the cell gets its energy, it gets its power to run around with the other reactions that are happening in the body. So let's describe more on the mitochondria and how it looks like in the next slide. So here's our mitochondria. We have repeated that it has double membrane, and with that we mean it has the outer mitochondrial membrane and the inner mitochondrial membrane. So meaning in between the outer and the inner mitochondrial membrane, we have a space here. But as we go inside the mitochondria, we are saying it has ribosomes. And so with the ribosomes, we understand the ribosomes are for protein synthesis. But also in the mitochondria, we have the mitochondrial DNA, and the inner membrane is also projected, or it has projections that you are calling the cristae. And we have the matrix in the mitochondria. This is like the fluid nature of the mitochondria, and this is where the respiratory functions happen, or generally the energy generating reactions happen to breakdown of the ATP to ADP. And ATP we mean adenosine triphosphate, while ADP is the broken down state of ATP. So when you break down ATP, you get ADP. ADP means adenosine diphosphate. And the other organelle is the cytoplasm or the cytosol. And the cytoplasm by structure, we describe it as jelly-like substance that is between the nucleus and the cell membrane. And its main function is to facilitate or to provide site for all the chemical reactions that will be taking place in the cell and its organelles. The other part is that it holds together the organelles in place or provides space for occupation of these organelles in the cell. And in our diagram here, The cell organelle is the cytoplasm, sorry, is the fluid part of our cell. So it is the part that has the organelles floating or positioned in. So it is the fluid in between our plasma membrane and our nucleus. So surrounding all these organelles, or rather bathing these organelles, is the cytoplasm. Other organelles, what we call cytoplasm, so not cytoplasm, but cytoskeleton. And the cytoskeleton, it's a group of filaments, or by structure, it's a network of protein filaments. And these filaments, we mean two types, that is microtubules and microfilaments. So these two microfilaments, these two filaments or protein filaments, they work together to support and give shape to the cell. So the main purpose of these two cytoskeletons is to give support and shape to the cell. However, you will find in the cell, they are widely distributed. You won't find them in one position. However, in these microfilaments, there's composition of two things. They contain actin and they contain tubulins. So the microfilament has actin, the microtubules has tubulins, and they work together to create a network for the purpose of shaping and supporting the cell. So as they support the cell, they generally prevent the cell from collapsing, if I can put it in that simple way. They prevent the cell from collapsing. And for this purpose, it therefore allows the organelles to have adequate space for them to function and carry out their daily activities or responsibilities. Smooth endoplasmic reticulum. The smooth endoplasmic reticulum belongs to the same family as the rough endoplasmic reticulum, as they all endoplasmic reticulums. But the difference is that the rough endoplasmic reticulum is nearer to the nucleus, and it also has ribosomes. Nearer to the nucleus, and it also has ribosomes. And the main function of the rough endoplasmic reticulum is for protein synthesis. However, for the smooth endoplasmic reticulum, the difference is that it is a bit away from the nucleus. It does not have ribosomes, and its main function is mainly to package the lipids and to break down any toxins such as drugs or alcohols. So what are the similar characteristics of these two is that the smooth endoplasmic reticulum is a thin folded membranes that are also connected together, but it does not have ribosomes, so as you can see here. However, it's just a continuation of the rough endoplasmic reticulum. So there's no ribosomes here. They're all folded together to form a stack of membrane or a connection of membranes. And the functions of our smooth endoplasmic reticulum is a transport pathway, what we are saying intracellular highway. So the smooth endoplasmic reticulum will undergo what we call lipid synthesis or formation of lipids, but also it will ensure there is breakdown of toxins or any materials that are harmful to the cell, and this can include drugs, alcohols, and other substances. So it's a transportation channel, but it's also a detoxification or a breakdown surface. So any material that are harmful can be destroyed within the smooth endoplasmic reticulum. But however, what you need to remember is the smooth endoplasmic reticulum does not have ribosomes. It is away from the nucleus. That's key for you to know. However, how does it ensure that all these functions are carried out smoothly? So we've said that the smooth endoplasmic reticulum focuses on lipid synthesis and destroying of all these unnecessary materials. So number one, because it focuses on lipid metabolism, what happens is the smooth endoplasmic reticulum will carry the following processes, or it will be involved in some of these processes. And one of them is lipid metabolism, cholesterol synthesis, and synthesis of the lipid components of lipoproteins. All this happens majorly in the liver. in the liver cells. So this tells you that in our liver cells, you'll find there's a boost of smooth endoplasmic reticulum for focusing or for mainly for this purpose of work. The other bit is the smooth endoplasmic reticulum works in synthesis of steroid-based hormones such as the sex hormones, and this can include testosterone, estrogens, and so on. Therefore, it goes without guessing that in the testicles or in the reproductive organs, there's a boost of smooth endoplasmic reticulum. It works together also in the intestines to do absorptions, synthesis, and transportation of fats. The other bit is detoxification of drugs, and here we have examples like the pesticides and carcinogens. The liver and the kidneys are the famous organs that conduct a lot of the drug processes and reactions, and if where there is need for destruction of drugs after they have completed the functions in the body, so the pharmacokinetics of the drugs happens a lot or happens actively in the liver and the kidneys. Therefore, smooth endoplasmic reticulum will therefore be in large number in these organs, the liver and the kidneys, for the purpose of destruction of used drugs or destruction of end products of drugs. And lastly, for the more functions breakdown, the smooth endoplasmic reticulum is able to break down stored glycogen to free glucose in the liver cells, especially. And remember, this is the one that can help us manage some of the conditions in diabetic patients, whereby when you break down glycogen to glucose, this leads to increase in glucose in the body and can promote diabetic characteristics. However, if you want to keep a client safe, then insulin normally comes in to just lower the glucose level, and this will include converting our glucose back to glycogen. But in the breakdown functions, it is the smooth endoplasmic reticulum that establishes these functions. And lastly, we have the organelles that we call centrioles or centrosome. This organelle, it's by structure, it's a small region of the cytoplasm that is made up of microtubules or a pair of centrioles. So if you look, the centriole, it's a pair of microtubules that are located on a small bit of the cell. And what the centriole do, they help during cell division. And in cell division, we mean mitosis or meiosis. And in mitosis, we generate two daughter cells with the same characteristics as the parent. In meiosis, we generate four daughter cells, same characteristics from the parent, but they have haploid nature of cells. So now, when you're looking at the centriole, how does it come in during cell division? So what the centriole does during cell division, they allow the microtubule to establish what we call polarity, or they establish poles. And that is, they establish one side of the cell to have a pole and the other end of the cell to have another pole. So that during cell division, they are able to pull our cell away from each other, create an equator, and then these cells can divide. But if you keep on with us on this channel, you'll be able to see as another class that will follow on cell division, and you will cover in details how the centrioles actually participate or actively contributes on the cell division, be it meiosis or mitosis. But for now, let's just appreciate that the centriole is a portion on the cell that will participate actively during cell division to help produce either diploid cells or haploid cells. So thank you so much for staying with us all the way to the end. Don't forget to drop a comment, share our videos, click on the bell so that you can be notified when our next class is coming in, and also remember to subscribe if you've not done. Like our videos. Thank you so much. My name is Winnie Barawa, and see you in our next class. Bye bye.